All right, yeah, thank you very much. As Luke mentioned, I'm going to talk about the research that I've been doing here at UBC, but I'm also going to talk a little bit, or actually quite a bit about the research that I've performed when I was at Cornell with Gaurav. And I want to kind of bring these two topics together in the end. Yeah, generally, I'm interested in large enzyme families, and then I'm also interested in how we can use these and manipulate them to use them in pathway engineering. First of all, the two places that I've been, 2017, I joined Gaurav's lab to basically study plant specialized metabolism in a little bit of a broader view on enzyme families. And since 2021, I am in at UBC Vancouver with Jörg and working on pathway engineering of MbB A, potential pharmaceutical for diabetic treatment. First, yeah, I'm gonna talk about these two topics, specifically about the BAHD acetyltransferase family, and then about the engineering of our MbB A by synthetic pathway. Generally, the plant specialized metabolism is characterized by a lot of diversity on different levels. We have huge amounts of metabolites. These are just a few examples of plant typical or plant metabolites. And people have been trying since for years to kind of estimate and to know how many metabolites are there in total. And the estimates, they vary hugely from around 200,000 to over 1 million metabolites. In this study here, they basically looked at unique metabolites per plant species, and then extrapolated that using the number of known plant species. And they kind of estimated that we could be expecting over 1 million of different plant metabolites. And unfortunately, or maybe fortunately, the same is true when we look at the enzymes that are responsible for this metabolic diversity. They are often part of large enzyme families that often emerge from primary metabolism, and after gene duplication get incorporated into specialized metabolism, and then through further repeated duplications and many of them lineage specific, we get a huge diversity and a large number of genes that are very closely related to each other. This is just an example. Probably have heard about this terpene synthesis, but there are many other examples for really large gene families. And this brings us to a very difficult problem. How do we find the enzymes that are responsible for our metabolic diversity if we have so many different enzymes? It's kind of like this picture here. It's a very complex network of different reactions, metabolites, enzymes. And to study this, I think most of us agree we need some sort of large-scale approaches and high-throughput approaches to decode this complexity, and then be able to harness this for the human. First, diving into the BAHD acetyltransferases. Most of you know this family. I'm just showing a very prominent example here from primary metabolism, which is the hydroxycinnamoyl shikimate transferase. This enzyme transfers coumaryl onto shikimate to produce coumaryl shikimate, and is essential for lectin biosynthesis and the synthesis of monolignol. This family has been studied quite a bit. There are many enzymes known, and people have found that they accept a huge array of different acceptors. They, for example, acylate long chain alcohols, flavonoids, but also alkaloids. All these compounds have very different chemicals, but as long as they have a hydroxy group or amine group, they're often acylated by BAHD acetyltransferases. The same is true when we look at the donors. They use a huge variety of different CoA activated donors, from aromatic hydroxycinnamoyl-CoAs to aliphatic CoAs with long carbon chains. We have studied the BAHD acetyltransferase family in terms of the question, when did this family actually expand in the plant kingdom? For this, we basically looked at 52 different plant genomes that have been sequenced from algae species to higher plants. And then we looked at the normalized gene count to account also for genome size increase. And what we found was that indeed the BAHD transfer family has expanded together with the emergence of land plants. But we see like 1 to 4 BAHD transfer copies in algae species. We see on average, 50 to 200 copies in higher plants. We then try to get an idea of what is the substrate space of these enzymes. And to do this, we basically went to the literature and tried to gather all the published BAHDs that we were able to find. We were able to gather 164 that were biochemically characterized, which is important because we really want to know if an enzyme is using a specific substrate, and we don't want to have just an association that could be based on genetic studies, for example, a knockout. We were looking at the substrates. We found 204 unique substrates, and then we organized them based on their chemical similarity to each other. We calculated maximum common substructure between those, and then organized them in this network here, and then classified them into different metabolic classes that are sometimes relatively similar to each other. As we can see here in this larger cluster in the middle, where we have different substrate classes close together, but then we also found very distinct clusters here, for example, which is sugar derivatives, or up here, anthocyanins and flavonoids. And then we went a step further, that we mapped basically these substrate classes back to the phylogeny. And then this resulted in this tree here. It's very small, so you don't have to see the details here. But basically on the outer side here, we mapped each of the different substrate classes to the different BAHD transferases here. And what we did then in the next step was that we inferred orthologous groups across our 52 plant genomes that I showed you earlier, and they using the software AutoFinder. And after inferring the orthologous groups, we were able to kind of estimate the breadth of conservation of each of those BAHD clades. I'm just going to show a few examples here and zooming in. For example, this clade here, 4A and 4B. These enzymes are mostly actinium coumaryl transferases, but also some other enzymes that all acylate amines. They produce hydroxycinnamoyl-CoA amines, which are widely distributed across the plant family. And also our approach showed that these are conserved between oil dicots and liverworts, so across the whole land plant family. They mediate tolerance to abiotic and biotic stress. And what you can also see that they have been extensively studied in monocots because they're very important in monocots. But most of these enzymes, they actually are from monocots, and without this orthologous group approach, we would have not been able to infer that they're actually conserved this broadly. Another example here, which is a little bit more restricted to lineages, but still pretty broad. These are anthocyanin and flavonoid acetotransferases. Those are conserved between oil dicots here, but also some of them are actually conserved across the whole angiosperm lineage. And this kind of matches very well with what we know about anthocyanins, that they are very important for flower coloration. The acylations give the ability to create different shades of color. And yeah, we also know that anthocyanins have not been found in non-land plants or even not in bryophytes. We also were able to kind of infer on a more broader scale when actually did the functional diversity of BAHD acetotransferases was established. Was this have all these activities basically evolved after they have radiated in land plants, or were there BAHD prototypes basically, or archetypes already present in the last common ancestor of land plants? And based on our analysis, it looks like that the last common ancestor of land plants was already, or had already BAHDs that were able to use all these different substrate classes. And for one of the substrate classes, which is the aromatic alcohols, or specifically here shikimate acylation, an important step in the phenylpropanide pathway. This activity we were able to find actually in streptophytic algae in a BAHD acetyltransferase clone from Cara. Next, we were interested in how the different clades basically evolve in different plant lineages. And for this, we basically selected a few representative species across the plant kingdom, from dicots, monocots, gymnosperms, mosses, and then we have bryophytes down here, and the algae species. And then we used two different approaches, which is BLAST and p-hammer, to map basically our clades to each of these individual BAHD acetyltransferase phylogenies. So we this is basically how it looks like. It's very small again. I'm going to zoom in a second, but I just want to show that there's kind of a big overlap between the two different approaches. We show here both approaches with no cutoff, which basically just take the best hit, which is probably prone to false positive. Then we apply a similarity threshold for BLAST, which is sometimes difficult because BAHD acetyltransferases diverge very quickly, and then the similarity actually plummets very quickly after duplication. So this could lead to a number of non-captured BAHD clades. And then p-hammer uses a hidden Markov model that we calculated for each clade. And then the software was able to map those clades a little bit more or more thoroughly, but in general, there's a huge overlap between both methods. Looking into this in more detail, we can see that certain clades have expanded in certain lineages specifically. One example here is clade 3 that has expanded in dicots. This matches very well what we know about clade 3. This clade is relatively diverse in terms of function. There are alkaloid acylazing BAHDs, there are acyl sugar producing BAHDs, and these are all chemistries that we find in dicots. We have another clade, as an example here, which is clade 4 that I showed you earlier with the acmatine-coumaru transferases. And this clade has indeed expanded in monocots specifically. And this mirrors kind of also the chemistry. The hydroxycinnamoyl amides were known to be very important in monocots and have been studied for this reason. Furthermore, clade 6, acylating aliphatic alcohols and compounds like terpenoids that are based on the chemistry relatively similar to each other have been expanded in gymnosperms, into the two species that we included here. And then we find a whole bunch of novel clades in ferns and bryophytes. I mean, this is also partly due to that that they haven't been studied that much, these clades, but also kind of interesting because there might be like some new chemistries or some new activities here that are not known. We also see always some novel clades in land plants that have not been studied, or we were not able to map them. Going back to this whole database, we can also use this to try and functionally or predict functions of unknown BAHD acetyltransferases. And here we combine kind of the phylogeny, the orthologous groups, and our substrate network to predict different functions. And then we basically validated them. And in the first step, we selected a number of enzymes that had not been characterized before, mostly from early diverging plant lineages because they hadn't been studied, and they were kind of like a proof of concept for our approach. If they basically map to a certain orthologous group, we predicted, okay, this enzyme must or likely uses aromatic alcohols, and then we try to recover that activity after heterologous expression and then in vitro characterization. One example here, I'm just not going to show everything, but like this enzyme here, this clustered within our well-known HCT and HQT clade from Achantia polymorpha, and it revealed that it indeed had the activities as acylated chickimate and kinase as like a classical HCT enzyme. Furthermore, 2 enzymes from Sphagnum, a moss, and Anthoceros agrestris, a hornwort. They're clustered in a clade down here that use aliphatic alcohols or long-chain aliphatic alcohols. And those 2 enzymes were actually acylating, for example, decanol and hydroxypermitic acid, and the ancestral, also the hydroxypermitic acid, using coumaryl-CoA, matching our prediction. Overall, we cloned a bunch of enzymes. Unfortunately, we were not able to express them all in active form, at least not in E. coli. But 6 of the 7 that we were assaying, we were able to correctly predict the substrate class. We then also tried validating our pipeline a different way from the beginning, and when we started the study to the point where we actually needed to validate it. There were new studies published on BAHD acetyltransferases, and in total, there were 28 newly characterized enzymes. And without these enzymes basically in our learning set, we tried to predict their substrates, and this was correct in the substrate class. This was correct in 23 cases, 82%. Another type of validation, which is a little bit more difficult to finally validate experimentally, is we try to improve the functional prediction for BAHD acetyltransferases from the tomato genome. In the latest version that we used there were 18 BAHD acetyltransferases that had some sort of substrate predicted or annotated to them. So basically, they were annotated as alcohol acetyltransferase. Some of them were computationally inferred, so they were not experimentally confirmed. But with our orthologous approach, we were able to, in combination with the already predicted annotations, we were able to bump this up to 42. Of course, this needs to be finally experimentally validated because only 9 of the BAHD acetyltransferases in tomato have known substrates. A different view on this whole thing is how can be made when we map BAHD acetyltransferase substrate to the phytochemical space. What is the phytochemical space? Relatively difficult because also we don't know most of it. However, we know many compounds, and there's a database called NAPSAC that contains about 7,300 metabolites with known structures that have been found in plants. And what we did, we basically mapped all the BAHD acetyltransferase substrates to this network. This network is again generated by sequence similarity. And what it shows is that BAHD acetyltransferases actually cover quite a nice range of substrates in here. So it's basically they have already explored quite a bit of the phytochemical space, but there are like thousands more substrates that are very similar to current BAHD substrates, which could be seen as like potential substrates for BAHD acetyltransferases that have not been discovered or that can be sampled during evolution. If we take out those that cannot serve as a BAHD substrate if they don't have a hydroxy or amine group. And 70% of BAHD acetyltransferases are actually not, or based on OGs, are not characterized. So this is also a way of maybe predicting what the remaining BAHD acetyltransferase is actually doing. Okay, now switching gears a little bit to the second part of my talk. And in a few slides that will hopefully become obvious, but this has to do with large enzyme families and also with BAHD acetyltransferases. The second part is about engineering the Montbreton A biosynthetic pathway in plant systems. Montbreton A, as I mentioned, is a potential anti-diabetic drug, and diabetes is a very big challenge. It's a global health challenge that basically affects 500 million people worldwide. It is the third most prevalent disease worldwide and under the top 10 causes of premature death. There are some treatment options already available for diabetes type 2, of course, in addition to dietary treatment by changing the diet. there are some medications that can alter the blood glucose level after the ingestion of starch-containing foods. There are just two examples here, acarbose and mictitol. Those are compounds that inhibit enzymes in the starch degradation pathway. Both enzymes, acarbose, for example, inhibits the human pancreatic alpha amylase, but also alpha glucosidases, and mictitol is only inhibiting alpha glucosidases. And what happens when you inhibit these alpha glucosidases is that you basically accumulate oligosaccharides in the gut, and these can be bacterially fermented and then cause in a lot of patients painful gastrointestinal side effects, which then lead to patient non-compliance. So patients don't take the drug anymore, so it's basically useless. And this was the reason why researchers have for a long time tried to find very specific inhibitors that just inhibit the human pancreatic alpha amylase to avoid these side effects. Here at UBC, in a large throughput screen of 30,000 natural product accessions, researchers were able to identify a compound called montbretin A. This was found in the corms of this plant here, which is Procosmia. This compound has been shown to be a very highly specific inhibitor for the pancreatic alpha amylase. It has already passed efficacy and toxicity studies in diabetic rats without showing these negative side effects. And it was also approved by Health Canada for phase clinical trials that are likely going ahead this winter or fall or winter. Previously, under Jörg Böhmann, Sandra Immisch and him have discovered the whole biosynthetic pathway of MbA. They found 5 UGT glycosyltransferases and 1 acetyltransferase, basically assembling this molecule. And this puts us now in a good position to basically try bioengineering in other host organisms, for example, microorganisms and plants. I'm gonna just show our approaches to engineer plant systems. We have for this bioengineering work chassis, we have picked Nicotiana benthamiana. You all know this plant and have probably used it in your own laboratories. It is being used also on an industrial scale to produce pharmaceutical proteins or vaccines. There was recently in Canada even a COVID-19 vaccine that was approved as Canada, unfortunately not by the WHO. But this plant can be transiently transformed at industry scale in these large vacuum chambers that you can see down here. It can be grown in these basically pink houses under artificial light. It grows very rapidly and has, in comparison to yeast fermentation, a very economic manufacturing cost. However, it has not been used so far to basically produce metabolites, more proteins. Why is this interesting in terms of large enzyme families? So there are several enzyme families that are important when we engineer this pathway in the plant. I'm just highlighting 3 of them here, 3 most important ones, which is the large family of the UDP glycosyltransferases. As I mentioned, there are 5 UGTs involved in our pathway, but then there are also over about 200 UGTs in the genome of Nicotiana benthamiana, and those are responsible for a number of undesired side products where we have our metabolite with the wrong glycosylation pattern. There are BAHD acetyltransferases. There's 1 BAHD involved in the pathway. We don't know if there are others endogenous to Nicotiana benthamiana that are interfering with our pathway, but this 1 BAHD, that is our pathway enzyme, is actually responsible also for the main side product that we see. And then there are methyltransferases, and methyltransferases are important because they're depleting our precursor pool that we need for our MbA molecule, and they are also basically indirectly responsible for the side product and likely low yield. I'm just going to go in detail for these 2 families here and what we have done to address that. This is a typical chromatogram, what you get when you purify after expressing the whole MbA pathway, expressing a Nicotiana benthamiana, and then you analyze this on the LCMS. And then you basically see two very prominent side products that are called MbB and MbC. And the only difference to our MbA molecule is that they have a chimeric acid moiety instead of the caffeic acid moiety or a ferulic acid moiety. And this is basically due to the fact that our enzyme here, the AT1, is relatively promiscuous in terms of its donors, of the donors that it uses. While we basically want that this enzyme uses mainly CaFeO-CoA, it also accepts very well chimeric-CoA, ferulic-CoA, and cinnamoyl-CoA. And we are approaching this problem with two different strategies. One is to modulate the availability of the substrate. And we want to do this because it has been shown in the past that BAHDs transferases is very challenging to make them very specific. Most of the enzymes that we know of and that I have been researching are promiscuous. It seems to be like kind of an inherent characteristic of these BAHDs transferases. Nonetheless, we're also trying to improve the specificity through enzyme engineering. This is pursued by Dr. Michael Eason in our lab. But I will show you a little bit about how we try to modulate the substrate availability for the enzyme. This is the basic phenylpropanoid pathway. Here we have the shikimate shunt. We have a bunch of different enzymes involved, all very well, most of them are very well characterized. Basically, we have here our three different precursors, our acyl donors that either lead to MbB, MbA, or MbC. And in an ideal, we would like to bump up the amount of CaFeO-CoA and reduce the number of chimeric-CoA and ferulic-CoA. However, this is quite challenging because this pathway is tightly regulated by the plant. It's very important for growth and development. So as soon as you basically interfere with this pathway, the plant will kind of react to the modulation of these precursor pools. What we have tried so far is overexpressing the hydroxycinnamoyl transferase here in a hope to like kind of shift the pathway to the right side here and to increase our CaFeO-CoA content. This actually works partly. We get way more MbBretons in total. We get more of our target compound MbA. However, we also increase the amount of our other MbBretons, our side products. Another strategy that we have tried is the RNAi silencing of an enzyme here, which is the CaFeO-CoA homicide transferase that methylates CaFeO-CoA to give ferulic-CoA. And there's another enzyme, it's the caffeic acid homicide transferase up here. Both enzymes basically give us this undesired acid group, the ferulic-CoA. By silencing this enzyme here, we were actually able to reduce or increase the ratio of MbA to MbC, as you can see here with three different RNAi constructs in comparison to a control without an RNAi construct. However, unfortunately, this also reduces the total MbBreton content. So we definitely have to do some additional work here to basically get to the same yields without the side products. And this basically brings me to my last slide, kind of the next step that we are doing in this project. We are currently attempting to optimize the MbA pathway flux by using different strength promoters within the pathway to kind of like eliminate the accumulation of any intermediate, which would then be available for the generation of side products. We are increasing the yield by modulating the expression of phenylpropanoid pathway genes, either by RNA silencing, but also by using transcription factors. And there has been like a recent study from the Sadley lab where they describe different transcription factors controlling monolignal biosynthetic pathway. and those are very interesting for us to use as well. We are also trying RNA silencing with UGT glycosyltransferases, and we don't expect like severe side effects of that approach, because most of these UGT they're not involved like in very primary metabolism pathways, so we don't think that the plant will like strongly regulate against that. We're also doing an RNA-seq experiment where we basically sequence strains that we have infiltrated with different combinations of our pathway genes, and combine that with the metabolomics experiment to identify new targets. Yeah, as I mentioned, we are performing enzyme engineering, and we are also exploring other plant hosts such as potato or tomato, which could then be stably transformed. And they have available agricultural cultivation systems, which could be beneficial for producing huge amounts of MbA. And with this, I would like to thank everybody who was involved in this research. I want to thank especially Gaurav and the lab at Cornell that I've been working with when studying the BAHD transferase family, especially my collaborators Austin and Divakar. Of course, I want to thank Jörg and Sandra for pioneering the MbA pathway work here at UBC, and all members of the lab that are involved. Our collaborators in UBC chemistry, Stephen Withers, and then also Knox Makunga at Stellenbosch University in South Africa, who is exploring cell culture production systems or plant cell culture. And then our industry partner, Plantform, Doug and Michael. Yeah, thank you very much. And